So I haven't broadcast yet. I'm going to wait. Oh, I'm going to broadcast now. It's exactly 8 o'clock. So good luck, everyone. Good evening, everyone. Welcome to the Friday evening lecture. We'll be beginning our lecture in just a few moments. Good evening, everyone. We'll begin the lecture in just a few more minutes.
Good evening, and welcome to tonight's Friday evening lecture. I'm Nipam Patel, Director of the Marine Biological Laboratory. I stand here in the historic Lilly Auditorium, home of the Friday evening lectures. Although the seats are empty tonight, you can see that we've been busy behind the scenes making audiovisual, safety, and cosmetic improvements. This work is made possible by a grant from the Massachusetts Cultural Council and a generous gift from the Millicent and Eugene Bell Foundation. We thank our funders for their support, and we look forward to welcoming you all back next year to a new and improved Lilly Auditorium. And now I'd like to introduce this evening's speaker, Neil Shubin. Neil Shubin is the Robert R. Bensley Distinguished Service Professor at the University of Chicago. He's a member of the Department of Organismal Biology and Anatomy. He also serves as a special advisor to the president and an advisor to the provost for the life sciences. Neil got his undergraduate degree at Columbia and his PhD at Harvard, and then went on to be a Miller Fellow at the University of California at Berkeley. Amongst Neil's many honors is being a member of the National Academy of Sciences, the American Philosophical Society, and the American Academy for Arts and Sciences. So Neil is famous for being a paleontologist. And his studies of paleontology have taken him to all 7 continents of the world, including most recently Antarctica. Neil and his team have discovered some of the earliest mammals, fish, and amphibian fossils ever found. And he's quite famous for the discovery of the Tiktaalik fossil, which I'm sure he'll tell you a little about today. But in addition to being a paleontologist, Neil's combined that interest in paleontology with really doing very cutting-edge work in the field of molecular biology, combining the study of fossils with the study of development. And he's been a leader in the field of so-called evolution and development. In addition to this, not only is Neil a prolific scientist, but he's a great communicator of science. So many of you will know his book, The Inner Fish. I think many of the public have read that book and been fascinated by the insights it provides. And many students have gone into the field of evolution and development because of that book. That book won him an award from the National Academy as one of the outstanding books when it was published. Neil's book, The Inner Fish, was made into a TV series, which actually went on to win an Emmy Award, and I think has been seen by many of you in the public. In addition, he has a new book, Some Assembly Required, where he again elaborates on the interconnectedness of studying fossils and molecular biology. So without further ado, I'll let Neil take it away. Thanks, Neil.
Well, thank you. It's a wonderful pleasure to be with you tonight, albeit virtually. What we're going to do is, from different parts around the world, different parts around the country, we're going to talk about 4.6 billion years of evolution and how it happens and some of the great transitions in evolution. One of the things that's always interested me throughout my career are the great transitions in evolution. How did fish evolve to walk? How did dinosaurs evolve to fly? How did bodies come about from single-celled organisms? And what we have now is just an unparalleled assembly of tools to analyze these great transitions in the history of life. I mean, it's really incredible. We have great fossils that we can go collect around the world. We have the knowledge of embryos and how bodies are built in developmental time. We have the knowledge of the genes that are involved in the developmental process and the way cells interact. So this is a really remarkable time to be studying the great transitions in evolution. So that all came together in a book I recently published called Some Assembly Required, using fossils and ancient DNA to understand the great transitions in the history of life. Now, as I was finishing the book, I was reading an autobiography of Lillian Hellman. And you can see Lillian Hellman here. She's famously hard-living ways. She was prosecuted by the House Un-American Activities Committee, McCarthy. She was blacklisted. She was a screenwriter and a playwright. She was blacklisted by that committee. And in looking back on her life, she was thinking about all the twists and turns of her life. She had a line in her autobiography which captured kind of how I view evolution, and indeed how evolution has been viewed since Darwin. And she said, looking at her life, nothing, of course, ever begins when you think it does. It struck me that's evolution in a nutshell. Nothing ever begins when you think it does. Things are always more ancient, whether it's genes or organs, and they always arise in a different context and are repurposed to play new roles over the course of evolutionary change. Now, what does this mean? What this means is if you think lungs arose as creatures evolved to walk on land, or feathers arose to help animals fly, You would be in really good company. Most people believe that. The fact of the matter is you'd be entirely wrong. And we've known that for over a century. And that's the key. Lungs, feathers, genes that control these things rise in one context and are repurposed for use in another. Okay, so let's look to see how this goes. So I'm a paleontologist, and I was originally trained to find fossils. And so what we did was I was interested in this transition from fish to land living animal, which you see here. A fish on top. It's a cartoon of a fossil fish found in rocks about 390 million years old. And the picture of a limbed animal on the bottom, a tetrapod, four-legged animal, tetrapod. That's a cartoon of a fossil found in rocks about 365 million years old. And what you see on top is, is a fish. It's got a conical head, has no neck, it has fins. And on the bottom, you have an early limbed animal, which has, limbs, as the name implies, has a neck, has a different shaped head, and all kinds of things. So what I set off to do as a paleontologist was to find an intermediate between that fish on top and early tetrapods on the bottom. And essentially try to find a flat-headed fish, with fins, maybe with arm bones inside. So what I did was, as a paleontologist, I was trained to think about, where do you look for new fossils that bridge gaps like this in the fossil record? Well, what you do is you look for places in the world that have three things. You look for places in the world that have rocks of the right age to answer whatever question interests you. Now, in this case, we're looking at the transition from fish to land-living animal. Remember, I told you the critter on top, is first discovered in rocks about 390 million years old. The one on the bottom, rocks about 365 million years old. So you're looking in that window of time between 390 and 365. It's the Devonian period, particularly the middle to late Devonian. All right, now if you're working on different transitions, obviously you'd work on different time periods. Okay, rocks of the right age. Next thing you do is you look for rocks of the right type. Not every kind of rock holds fossils. Some are, formed from lava, magma. They're not called fossils, or nothing's going to be in there that was once alive. Metamorphic rocks that are highly pressurized and heated up, those aren't going to hold fossils either, or if they do, they're going to be grossly deformed. So what you do is you look for sedimentary rocks that are from the environments that those creatures you're looking for likely lived in. In this case, we're looking for things that lived in nearshore environments, rivers, streams, things like that. So you're looking for sandstones, siltstones, maybe shales that would contain these. Finally, it does me no good if my wonderful rocks of the right age and the right type are buried, 5 miles underground. Right? They have to be accessible at the surface. So accessibility exposure. So that's what we do. We apply those filters: rocks of the right age, rocks of the right type, rocks that are exposable. We apply those filters. And all of a sudden, a giant globe becomes much smaller. You have a candidate list of places to look. And that's essentially what we did as we started the hunt. Now, what we realized early on, that the right environment for us to find one of these creatures was likely, the one that would maximize our chances of success, was likely ancient delta systems, much like the Amazon Delta today. And you can see here, this is actually a reconstruction of some Devonian delta systems. This is one actually in Pennsylvania. What you see is highlands on one side, an inland sea to the other, and rivers that drain from the highlands into the sea. Now, this is great because if you have this kind of setting, and you have rocks of the right age that are exposed in this setting, all of a sudden you're maximizing your odds. Because if you have the right exposures, you can sample ancient nearshore environments, estuaries, and so forth. You can sample ancient rivers and streams, ponds, and the like. So really, what we're looking for is ancient delta systems preserved in rocks between 390 and 365 million years old. Actually, got a little more refined than that, and ones that were accessible. Well, our hunt led us to the Canadian Arctic. Since then, it's led us elsewhere. But what you can see here in this slide is the Canadian Arctic, particularly Nunavut Territory. Now, Nunavut Territory is the northernmost province of Canada, and you can see where we are in the upper left of the slide. You can see the North Pole here, and Nunavut Territory circled in red. Now, the main body of the slide, what you see are the islands of Arctic Canada. And these islands extending from Ellesmere Island in the east to Melville and Banks Island to the west, you can see circled in there in red are where Devonian age rocks, those rocks of the right age, formed in ancient delta systems, rivers, streams, and marine environments, were preserved. And so this was the ultimate place for us to look. And you can see it's really vast. And it was really just a matter of getting to those sites to be able to find the fossils. So we started this project in 1999. And what you see here is our first camp in 1999 in the western part of the Arctic. Excuse me. And you can see what camp looks like. You know, we each have our own personal tents, we have a kitchen tent. But you can see here, those in the distance, those are the Devonian rocks. And one thing that should strike you immediately is how low-lying that part of the Arctic was. It wasn't ideal to find fossils. There are two problems with that first season. We were in rocks of the right age, but we probably were in rocks of the wrong type. These were marine rocks formed in ancient oceans, but they weren't near shore. They were like in the middle of the ocean. So we were really in the wrong place. So that first year, lessons learned, we learned how to work in the Arctic. We learned where not to look in the Arctic. So back to the drawing board. So back to the drawing board meant, and let's go back to the diagram here showing the delta system. What you see is, well, we're in the middle of the ocean. What we needed to do was go upstream. So when we looked at the geology, going upstream in ancient Devonian rocks in the Arctic meant going east. So that's what we did. Here in the next slide, what you're seeing is where we were in 2002. And here you'll notice a couple things. We are in a much more montane environment. Look at across the fjord, see those cliffs there. That's all Devonian rock. And it's all Devonian rock, the right age, formed in ancient rivers and streams. So we really were getting closer here. This valley, though, these rocks, though, were not ideal in the sense that these were rapidly moving streams. So whatever fossils were in there in the Devonian would be broken apart by the currents. We needed a place that had slower moving streams or margins of streams we can look at. So we went to this valley here, the next slide, and this is near perfect for us. Because what we found in this valley, and you can see in the center of the slide, there's a big hole we dug. That hole we dug at a layer of rock that contained, you know, one skeleton of fossil fish piled on top of another. In fact, there's probably thousands of skeletons of fossil fish. So what we had now was a site in rocks of the right age and rocks of the right type, rocks of the right age. We had a site that was relatively compact, and what we had to do was remove the rock on the surface of that site to expose the skeletons inside. And that took us about another year. I'm basically going through 6 years of work in a small number of slides. We worked next to one another, and everything changed as we were removing rocks when a colleague pulled a rock from here, and you can see that in this slide here, and he pointing to the center of the slide. He said, "Hey guys, what's this?" And I looked at that, and I realized that that is exactly what we were looking for. That I was looking at not only the head of a fish, but because it had fish ornamentation on it, but it was the head of a flat-headed fish. You know, so flathead, remember I said we're in the sort of the tetrapod area there. That's what we're looking for. And let me show you the slide. So if you look at the slide, you don't really see much, but here's where it is. If I look, you can see the arrow is pointing to the tip of the jaws here. That was the upside down skull of a flat-headed fish. Yeah, so it's amazing. So we brought that back, wrapped in plaster, and we since found 20 more of these individuals of these flat-headed fish, some skeletons, some individual bones. So that specimen, you know, comes back to the lab, and then a fossil preparator sits with a needle and a pin vise to remove the rock grain by grain. We now do that. This is 2004. We now do this by CT scanning. You know, high energy CT scans can blast through and really you can remove, prepare things digitally. But at the time we were doing it manually, and you can see here, he started to remove the rock, and look at that. You can start to see, look, it's a flat head. You can even see sort of two orbits or eye holes. It looks like we're looking at the dorsal surface of a skull, the top surface of a skull. Okay, and a few more months go by, look at this. All of a sudden the head starts to reveal itself, flat head, eyes on top. Looks like this thing has a neck, because you can see shoulder blades back there. Looks like we're in the right ballgame. So, let's go back. We're looking at fish on top, early limbed animal on the bottom. We're looking for a flat-headed fish with fins, with arm bones inside, and this is that exact specimen. This is the specimen that was in the rock that I showed you before. Here it is. If I was to hold it in front of you, it's about 4 feet long. The biggest one's probably about 10 feet long, so we have a range of body size. It's likely a growth series of these things. In fact, now we found even a smaller one. You can see flat head, eyes on top. Look at it, scales in its back like a fish, has fins with fin rays like a fish. You can see the foreground left here. But it has a flat head with eyes on top like an early limbed animal. It has a neck. And in fact, when we crack open the fin, what do you see? You see bones that correspond to upper arm, forearm, even parts of the wrist and stubs and digits, and a fish fin. If we look at the phylogeny of the cladogram, this is kind of the evolutionary relationships. What you can see is the sequence of the time of critters at the cusp of the transition from life in water to life on land, going from a fairly fish-like thing with fish-like proportions in the skull, with fins and so forth, all the way up to tetrapods at the top. And this critter, Tiktaalik rosea, is right in the middle there with a sort of a mix of these features. In this slide here, what you see is the fin. And what's really cool is when we look at the fin, it has bones that correspond to upper arm, forearm, parts of a wrist. It's a fin that has palms that can articulate and touch the surface. It's a fin that's set to do a push-up to support the body. So what this shows is that many of the features that tetrapods were used to walk on land originally rose in very fish-like creatures. And in fact, we can even take it deeper still. Arms, wrists, legs, things like that were originally found in fish living in aquatic ecosystems. So nothing ever begins when you think it does, in a really important way. And that approach really applies very importantly to understanding molecular biology and how genes evolve over time and how developmental processes evolve. So this slide shows kind of where it all begins, right? Fertilized egg. For animals, a fertilized egg is really important because it contains DNA, it contains all kinds of maternal factors, proteins, and other things that are inherited from the mother inside the egg. And what happens over time is from that single cell, a body emerges. It's really amazing. And so we call this process, in the next slide here, bodybuilding, going from a single cell to, a 4 trillion celled individual or creature, as you see on the right. That's just remarkable how you have a sort of a DNA toolkit inside the cell with maternal factors, with environmental influences throughout development, that's read out as cells divide and differentiate to make organs that are all packed in the right place. This captures sort of the beauty of development. Understanding that process has turned out, as many of you know, very critical to think about evolution. And in fact, it even gets more interesting when we think about the DNA inside the cell. And think about this for a second. Here you have DNA wrapped in a nucleus. In every single one of our cells, we have about a 6-foot-long strand of DNA composed of about 3 billion bases, all wrapped and packed in on itself inside the nucleus of the cell. If you were to take all the DNA of our bodies, of all the 4 trillion cells, each 6 foot of them, lay them end to end, they would go from here almost to Pluto. It's a remarkable amount of genetic information, computational information, if you will, to build a body, to keep a body thriving. And in fact, the more we learn about how the DNA is packed inside the nucleus of the cell, and the more we learn about how dynamic that is as it opens and closes and changes shape, the more we're understanding how genes are turned on and off in development and what controls that process, as bodies are built and as, species evolve from one to another. One of the great discoveries that's really informed my own research as well as, a whole field of research, in the last several decades, in the last 3.5 decades, has been the realization that there are a number of genes that are active, and genetic networks, I should say, that are active during the building of bodies. You can see here's a human embryo and a human body. We have a basic body architecture where organs such as limbs and backbones and vertebrae and skulls are in the right place, articulated with one another. And genes are turned on and off, obviously, during development as these organs form. And it turns out many of the most fundamental genes that build organs that are, that control the development of our organs are also seen in other creatures, such as here, fruit flies. And versions that just was when that insight came out back in the in the late 1980s, early 1990s, it was truly mind-blowing for so many of us in many different fields, because we're beginning to see that versions of the same toolkit that build the bodies of creatures as different as flies and worms and mice and people and fish have sort of universal qualities to them. That genes can arise and developmental processes can arise in one species, in one context, an organ, and then redeployed for other organs as well. And really kind of is very clear when we look at these kinds of genes, and I'm going to be looking at genes called Hox genes from the Hox gene family. It's, again, this is a gene family that extends from, was originally studied in flies. We now know they're active in mice and people. But they're not only active in the building of the basic body axis, which you see here, but they're also active in the development of other organs. They've been co-opted to form genitalia, limbs, and other structures. So in this structure, I'm going to talk a little about limbs and what the role of these genes are in limbs. Because again, it's going to come back to Lillian Hellman's quote, nothing ever begins when you think it does. So here's a limb. All right, when you think about what our limbs are, appendages, we have upper arm, forearm, wrists, and digits. Well, they have a specific name, and we color-coded them here, red, yellow, and blue. The names we give them are stylopod, zugapod, and autopod for those 3 regions, upper arm, forearm, and parts of the wrist. 1 bone, 2 bones, little bones, fingers. Well, we can see everything that has a limb has that pattern, whether it's a bird, whether it's a whale, whether it's a frog, whether it's a person, whether it's a mouse. 1 bone, 2 bones, little bones, fingers, stylopod, zugapod, autopod. Question is, when did those compartments come about, and what controls their development, and how did that evolve? Well, when we look in the fossil record, what we find is in the Devonian period, around 365 million years ago, we already have limbed animals, okay, which have one bone, the stylopod, which have two bones, the zygopod, and which have wrists and digits. Well, does this extend in fish? Well, if we look at Tiktaalik rosea, the creature I was just showing you, as well as other cousins like it in the fossil record, what we find is a real mix. What you have is a stylopod. It has a humerus as well as a femur and leg. It has radius and ulna, the zygopod, and it has distal elements which actually conform to a portion of a wrist, all set in a fin with fin rays. See, what you see here is the fin rays shown in black and white. Those are formed of a dermal bone, a different kind of bone, and the bones that correspond to our wrists and digits and so forth actually lie within a sandwich of those bones, fin rays. But if you compare these to, say, a zebrafish, a living teleost fish, there really are no morphological anatomical markers that us allow to compare among these. It's a little more challenging. Do fish have versions of stylopod, zygopod, and autopod? Do we share anything with them? And the answer to find answers to that, what we've had to do along with other laboratories is to really think about development, to think about how these things form. So here you look, there's a fish on the left, a human on the right. And you can see, you're looking at the fin on the left, and you're comparing it to a human arm on the right. There's kind of like not a whole lot you can compare, right? The fish fin has fin rays. We don't have fin rays, like all tetrapods. We have the stylopod, zygopod, and autopod, and there's no obvious stylopod, zygopod, and autopod in fish. I'm showing you a zebrafish here, but it would apply to other fish as well. But look at development. And let's think particularly about those Hox genes I told you about, the ones that we share with flies and so forth, what they're doing in development, because they have proven to be very powerful markers and powerful insights into powerful mechanisms of evolutionary change. So here you see, this is a mouse appendage, and on the left is a developing mouse limb, and you can see it has this little paddle. And on the right, stained in blue, are the bones of the mouse limb, a newborn mouse, and you can see its appendages. You can see the digits and the wrist and so forth. Now, what's in black here are where HOX13 genes, now there's several different kinds of different flavors of HOX13 genes, where they're active during development. And what you notice is when you look on the left, that whole paddle area of the mouse limb, which is going to form the wrist and digits, is where these genes are active late in development. Well, these, it turns out, are really wonderful markers for that autopod, that is the wrist and digit area, in humans and mice, in fact, all tetrapods. Because if you look at this, if you label these genes, and this was done by Marie Comita's lab in Canada, if you label these genes with a permanent marker that allows you to trace where the cells that have their activity, where they end up in the adult, check it out. What you can see in green here is genes that had expressed or were active with HOX13 genes, in this case, particularly one of them, HOXA13. What you can see is it marks the wrist and digit area nearly perfectly. So a beautiful marker these genes are for the autopod. Furthermore, some labs back in the 1990s and early 2000s made mice lacking these genes, lacking their activity in limbs. And what happens when you have a knockout of these things, what you do is when you remove these genes from the development of limbs, what you end up with mice that have one bone and two bones and then no wrist or digits. Okay, so but in the lineage tracing sort of environment and the gene knockout environment, what you have are these HOX13 genes being really wonderful markers for wrists and digits. Well, I'm an evolutionary biologist, and I'm a paleontologist. I'm asking, what's happening in fish? Do they have these genes? The answer is yes, they definitely have these genes. But what are they marking in fish? And is there a similar region in fish fins and tetrapod limbs? And will it tell us about how limbs arose from fins? Well, if you look at, here's a mouse, and here is the gene activity of two of these HOX13 genes active in limbs. There's a phase of activity, so shown in black here is where they're active in this stage of development. When they're both active at the stage of development where digits and wrists are specified. See that paddle area there is where the wrists and digits will form, and that's where these genes are active in late development. So it's discovered both in Cliff Taven's lab at Harvard and Tanita Poole's lab in Switzerland. a number of labs, including my own, started to say, okay, what's the pattern of activity in fish fins? And it was really suggestive. So if you look at work done on paddlefish in my laboratory, what you'll notice is that there's a terminal strip of tissue that has this sort of late phase, I'll call it digit-like activity in paddlefish. And if you look at zebrafish, the same sort of thing. In the terminal end of the fin, there's a terminal strip of tissue that would seem to compare to the autopod or the wrist and digits of mice. So that was really suggestive to us that perhaps that there's a terminal patch of tissue in a fish fin is comparable in some ways to the terminal region of a tetrapod limb, allowing us to compare limbs and fins. So what we tried to do then is to replicate some of those experiments with lineage tracing and knockouts that I showed you for mice. And here I had some really remarkable people in my laboratory. This was about 6 years ago when we sort of launched these studies. This is Andrew Gerke, who's a graduate student in my lab. He's now in Nancy Shubin's lab at Harvard working on regeneration. Andrew was really passionate about really thinking about how to do lineage tracing and gene regulation of these genes. I'm not going to talk about the regulatory story, which is a whole other chapter of this. We'll talk about the lineage tracing. What he did is he used a cell marker line developed from Len Zhan's lab in Mass General, where you can use a ubiquitous switch marker to label cells permanently as they express HOX13 genes early in development and map them all the way to the adult. And this is what happens 6 days post-fertilization. Here you can see the fin, and shown in red here, reddish pink, are the HOX13 expressing genes. And look at them, they all of a sudden start to fill the terminal space of the fin. And in fact, if you follow them even later, 21 to 3 weeks post-fertilization, look at this. They've already migrated and started to form the fin rays. So it began as a terminal strip of tissue in fish fins expressing HOX genes, now reveals cells that are migrating and dividing terminally to form the fin rays. So when Andrew looked at this, around birth, what he found is that you can overlay, bone markers with late phase activity, and it really does seem that these the cells that express this HOX activity are in part osteoblast cells, bone forming cells that form the fin rays. So the end result was to say, how can we use these HOX genes to compare fins and limbs? Remember, I showed you that the HOX13 genes form in lineage tracing environment beautiful markers for wrists and digits. If we do the same experiment in fish fins, it's also the terminal end of the fin, but in this case, it's not digits, it's fin rays. All right, that was, as a paleontologist, that was a bit of a surprise for me. I was not expecting that. I was expecting really kind of the comparator to be the cartilaginous bone. Okay, then I had somebody else remarkable come into the lab who did the other component of this. This is Tsuya Nakamura, who is now has a lab of his own at Rutgers University. Tsuya is no stranger to the NBL. He worked on skates here, as well as worked with Jose Luis Gomez Scameta, a colleague of mine, doing a tax-seq of different fish. So Tsuya learned cribbed techniques on CRISPR-Cas gene editing to do knockouts from Marco Horb at the Xenopus facility. Also Jonathan Gitman, who was here at the time, who was doing zebrafish CRISPR-Cas knockouts. So learning from them, he designed a knockout strategy to knock out HOX13 genes in various combinations. Remember, I told you there are a number of them, 2 sets of them active in limbs. So what he did is he engineered HOX knockouts using CRISPR-Cas. This is back in 2013, 2014. And what he did is, I'll cut to the chase, I won't go through all the details of the experiment. What he did is he was able to generate knockout phenotypes and then use our paleontology CT scanner, the one that can really blast through appendages, to get detailed phenotypes and anatomical patterns for the fins. What it did is it allowed him really to look in great detail, what are the anatomical effects of knocking out these HOX genes. Now remember, what they do is when you knock them out in mice, what happens? You lose the wrists and digits. What happens in fish fins? This. And again, cutting to the chase, there's you have to knock them out all together in one combination. In the wild type, what you find is in the proximal or base region, where you have our bones that are formed out of cartilage, so in the dark there, and then the terminal end, you have the fin rays. What happens in the HOX knockouts on the bottom? What you find is the cartilaginous bones are largely the same, but you've really knocked out and reduced the fin rays. And in fact, when he looked at them, there was another puzzle that emerged, though. If you look at the cartilaginous bones at the base of the fin, What you'll see on the left is the wild type, right, the untreated one, and then you can see there's two sets of these bones. One's in gray and the one's in red. The ones in red are what are called the distal radials, and there's a set number of those. There's usually about 5 or 6. In the knockouts, remember we reduced the fin rays, but in the knockout individuals, we actually increased the number of the anaconda bones, of the red bones. So it's really, so we're increasing the cartilaginous bones, decreasing the fin rays, the dermal bones. So if we look at this in the mouse, what we have is late phase expression, right? The markers for the autopod. In the lineage tracing and in the knockout environment, what do you have? You have markers for the autopod. Well, do we have an autopod developmentally and genetically in fish? Well, it looks like the comparison is a terminal strip of tissue, which has that late phase expression, and then the fin rays, largely the fin rays here shown in the lineage tracing as well as in the knockout environment. So what's going on? Well, what we think is that the digits and the fin rays are comparable in some important developmental sense. Doesn't necessarily mean they're homologous in an evolutionary way. What it means is that they're formed from similar populations of Hox-expressing cells. That is, if you look at the top here as a mouse, you have Hox-expressing cells shown in red, right? Forms as a distal strip of tissue originally, becomes that whole paddle region. Those are the cells that are going to give rise to the wrist and digits. Look in the middle, what happens in zebrafish is you have a terminal, little smaller patch of tissue, it's a strip of tissue. Those cells will migrate and form the fin rays. What happened in our mutants in the bottom is we think we blocked that migration or blocked their proliferation in some way. That's what caused the defects that we saw. So what we have is a terminal strip of tissue that expresses versions of genes that were found originally discovered in flies, but shared with mice. That distal strip of tissue forms the fin rays in fish, forms the digits in tetrapods. Nothing ever begins when you think it does. When you think about the subtle shifts that have to happen in development for those tissues to acquire new behaviors to go from forming fin rays in fish to digits in tetrapods. So when you think about the fish to tetrapod transition, life in water to life on land, that's a huge transition, right? I mean, so much has changed. Fins to limbs, necks have to arise, lungs, all kinds of physiological systems. But remember, nothing ever begins when you think it does. I'm going to show you that much of what tetrapods needed to walk on land already arose in fish living in aquatic ecosystems. And the story takes us to one of the, I'm jealous of this guy, Bashford Dean. He had two of the greatest jobs, like in the history of jobs. He was curator of fossil fish, of actually armored fossil fish, at the American Museum of Natural History in 1910, 1920s. He also, at the same time, held curator of armor, of medieval armor, at the Metropolitan Museum of Natural History. He's the only person to hold joint curatorships at the Met and the American Museum of Natural History, right across Central Park from one another. And in both cases, he was studying armor. He was studying, the medieval armor, and actually samurai armor and so forth at the Met, but he was also studying armored fossil fish at the American Museum of Natural History. And he loved to wear his armor on the streets of New York. What a character. He was a really remarkable person because he was interested in fish, but he was also interested in fish that breathe air. And so of the fish that captured his imagination, lungfish were particularly important. Now, lungfish are alive today. They have an extensive fossil record. But they're alive today. They're alive in the southern continents, the Gondwana continents of Australia, Africa, South America. This is the Australian lungfish, arguably one of the cutest fish ever. Look at that, it's adorable. But they have lungs and gills. And what that fish does is it will breathe air. So as Dean realized, what it does, it lives in streams that have variable oxygen content throughout the year. When air is relatively abundant in when oxygen is relatively abundant in water, they can get by mostly with their gills. But when the oxygen content drops, they tend to breathe more. What they'll do is they'll go to the surface and they'll actively gulp air and force it into their lungs. And looking at their lungs, what Dean realized is that they have a paired set of lungs, and those lungs have alveoli. Those lungs have surfactant. In fact, what we know now is that the genes that form the lungs of lungfish are similar to the genes that form our own lungs. And indeed, when Dean looked at the distribution of lungs in fish, he suggested that lungs were primitive, that lungs arose well before animals took their first steps on land, and an observation that's only been confirmed in the years since Dean. So here you have a phylogeny, a cladogram, going from armored bony fish at the base to sharks and cartilaginous fish, and then rayfin fish of different kinds, lungfish, and then tetrapods. If you look at the distribution of lungs, it appears that lungs are found in basal rayfin fish, they're found in lungfish, they're found in tetrapods, and indeed there's even some suggestions, although controversial, that they're present in basal armored fish as well. But regardless of whether that's true or not, lungs were around well before the critters took the first steps on land. So what do we have? We have, in the fish to tetrapod transition, it's not like critters had to, wait for all kinds of new features to arise so they can walk on land. No, what they did was creatures living in aquatic ecosystems of the Devonian evolved wrists, digits, portions of digits, arms, legs, lungs, and so forth to live as fish living in these aquatic ecosystems, such that when the environmental conditions changed or the opportunities opened up, they already had the tools to walk on land. They were ready to go. And this is not just a one-off in this transition. This, nothing ever begins when you think it does. It's true with pretty much every great transition in the history of life. I mean, if you take the origin of birds, and here's a beautiful Mandarin duck. What do they have? Feathers. They have hollow bones. They have wishbones. They have wings. All high metabolisms. They have all number of features that, if you were to think about, how birds would arise, you'd think it would be impossible because of the number of features that would have to arise simultaneously. The fact of the matter is they didn't. Those features came about sequentially and gradually in dinosaurs running on land. The feathers that arose were originally seen in dinosaurs. They're not used for flight. They were used for probably courtship displays or thermoregulation, but they clearly weren't used in flying animals. These fast-running carnivorous theropod dinosaurs, they had hollow bones, high metabolisms, evolved some sort of wishbone, such that at some point they already had all the tools, evolving as dinosaurs living as carnivore, fast-running dinosaurs living on land. They already had the tools to take off and fly. So that's the story of evolution. It's true for genes, it's true for organs. Nothing ever begins when you think it does. Things are continually arising in one context, redeployed, co-opted, and repurposed in a new one. So we come back to Lillian Hellman. So she's just, she captured it all with that quote. Now I'm continuing thinking about these ecosystems. Obviously, we have tectonic rosea, but there's rocks that are younger, there's rocks that are older. I mean, really thinking about not only the transition itself, but the environments that these transitions happened and what did the world look like at this time. Turns out, so when we think about rocks of the right age and rocks of the right type and places to look, one of the best places to look is one of the ones we've been working most recently, about a year and a half ago, is Antarctica. Antarctica, as you know, is a vast continent, larger than North America. Mostly it's an ice sheet, but there are mountain ranges that poke through Antarctica. And those mountain ranges contain rocks of all kinds of different ages, including Devonian age rocks formed in ancient rivers and streams, delta systems. So we've led 2 field seasons down to Antarctica. Year 1 seen on top to the area near what's called the dry valleys, and then year 2 in the bottom in what are called nunataks, which are mountain ranges that poke through the ice plateau. The ice plateau there is at about 5,000 feet. These mountains that poke through go up to about 7,000 or 8,000 feet. So that gives us about 3,000 feet of mountaintops to work on. We camp the base and then the ice and then hike up. This is just an incredible place to work. This is from a helicopter recon. These are some of these mountains that poke through the ice. It's imagine like Grand Canyon with glaciers. That's what you got. Now, if you're a paleontologist, these kinds of exposures are really fabulous. Now, it's a challenging place to work. This is like my commute to the morning coffee. Those are the kitchen. There's a science tent on the left, and then a kitchen tent on the right. You get around with snowmobiles. What you see in the distance there are the mountain ranges of the Devonian rock, and they're sticking through the ice plateau. This is New Year's Day, 2017. Yeah, so that it does get down to -30 and does snow a lot. The snow is really dry, but we don't have heat. The only heat we have is like what we have in our bodies. We do have propane stove, so we use that to cook. And I actually can bake bread in that setting. Believe it or not, I'll give you my recipe another time. But we work on these rocks, and what you can see here is one of the areas that was actually pretty productive. Look to the base of this diagram, this photo, and you can see what's white there. This is a shale that actually was a layer of shale that had fossil skeletons of fish inside it, and some of them were relatives of Tiktaalik, actually, that we're still working on. Other areas were really challenging. So, for instance, this one here is an entire pavement of fish bones, skeletons. That we're beginning, we're a year and a half into it, we're still taking it apart. And that was a plateau of bones, of skeletons that were piled one on top of the other, so much so it's hard to tell the distinction between one individual and the next. And so that's why we're using CT scanning technology to begin to take it apart. Because if we did it destructively, there's no way we'd be able to put it all back together. So we're in the process of making sense of that. Some really remarkable stuff has come out. Yeah, it's another slide. So there's a lot of these ancient armored fish. Remember I showed you that some people claim that they actually had lungs. These are armored fish that sit at sort of the base of the tree of bony fish. And they've been argued about, are they bony fish, or are they primitive jawed fish, or whatever. They have some of the earliest known bone in the fossil record of our type. And it's an exoskeleton armor. That's why Beschrutine loved these fish, by the way. We find a lot of these. And so here's one, but we find a special kind. This is one you can see you see the front is on the left, the back is on the right. You can see that it's the spine of its fin is on the top. What's remarkable about these is we have larvae of these as well. So it's not only do we have whole bodies of them, but we have what appear to be growth series of them. So we'll be able to look at the development of some of the earliest bone in the fossil record. So Antarctica is an ongoing project, but we're really excited by the amounts of what we're finding. It's not possible to get there now, but we have enough to keep us busy for quite some time. So when you think about evolution, when you think about how we study evolution, this is an embryo here. We could study evolution by understanding genes, manipulating genes, thinking about gene networks, thinking about how cells interact to form organs at all levels of biological hierarchy. We could begin to do that in a comparative way to understand how different organs compare to one another in terms of their genetic similarities and their developmental similarities. We can go out around the world and collect fossils from the Arctic to the Antarctic. We can look at all 7 continents to begin to see fossil intermediates. And what we discover when we do that is nothing ever begins when you think it does. Structures, organs, genes, gene networks always arise in one context and are co-opted and repurposed to form new ones. And that's the creativity and beauty of 4.6 billion years of evolution. So thank you. The work that I do could never happen without some really remarkable people who I've had the chance to collaborate with. I've talked about Tsuya Nakamura, Andrew Gerke, 2 people whose work factored in this talk who I didn't mention were Justin Lemberg and Tom Stewart, 2 postdocs in my lab. I had the great privilege of working with some co-PIs in the Arctic project, Ted Deschler and the late Ferris Jenkins. Been a member of the US Antarctic program. I should give a huge shout out to Jose Luis Gomez Scarmeta, a member of the NBL committee who really jump-started my work on gene regulation. Which I didn't present here, but it's been a major part of my own work. And of course, the funders. So thank you very much. I'm happy to take your questions.
Hi, Neil. Thanks for that great talk. So I want to just start off by, again, thanking everyone for attending. We wish that, we could have everyone here in the Lilly Auditorium, but we've had great attendance, and it's been wonderful to have sort of the alumni of the Friday evening lectures come and give us talks. And so I think questions are coming in, and so let me go ahead and start, Neil. So the first question here is, how do you identify Devonian land features like deltas and upstream locales where your fossils were found?
Yeah, that's a great question. And so we rely very heavily on the work of sedimentologists and geologists who map the Arctic regions decades ago. So one of the things that happened in the 1960s and 1970s was during the Cold War, the Canadian government put an enormous amount of resources into mapping the rocks in the Canadian Arctic. And they were interested in, oil and gas extraction, mineral extraction, but also there were land claims and other things going on there. But what they did is the teams that did that did the basic geological mapping that's necessary to find fossils. And so what you do is when you can tell the marine rocks formed in an ocean, there's sort of black shales and things like that, differ from rocks formed in rivers and streams, which are more conglomerates. They have pebbles and small particles in them and so forth. So you can use sort of sedimentological analyses of individual layers and whole units to understand, whether you're in an ancient stream or whether you're in an ancient seaway or an ancient estuary and so forth. Sometimes it can be kind of tricky, but at the extremes, it's not. I mean, you can definitely tell when you're in a river or a stream geologically versus an ocean. Sometimes it can be tricky to tell whether you're in freshwater or saltwater. That can be challenging. You look at the plants that you find there. You look at the fossils you'll find there. You may look at some of the geochemistry of the rocks as well. So what you try to do is pull together as many tools as possible to do that. We were fortunate in the Canadian Arctic, as well as in the Antarctic project, that geologists had been on those sites before us and had mapped them and done some really careful work in advance. And that was sort of the seed corn for our own work. We could never have been there if it wasn't for that basic science investment that, sort of that told us essentially where to look.
Here's another really interesting question I think that probably people will be fascinated by. What is the evolutionary advantage of having arms and fingers when you're still a fish?
Yeah, so when you think about where these fish are living in shallow streams, most likely, often choked with weeds and logs and so forth. So they're maneuvering in a very complex environment. So the ability to walk on the bottom of the water, to maneuver through complex landscapes, probably began in water. Okay, we know that. We know lots of fish have alternate gaits when they walk on the bottom of the water. And they have, some living fish today, have their own version of a little palm that they use to maneuver about. What fingers and arms do is they're a way for the appendage to support the body as the critter moves around, right? But think about these environments as sort of a continuum. What you have is a continuum from the water bottom to the shallows to mud flats. So the same traits that would allow a creature to walk on the bottom of the water would be useful for walking in shallows as well as on the mud flats. So think of the walking in water as kind of the training wheels, for the creatures to walk on land. So that environment was actually rich in a lot of things because, once you have creatures supporting themselves with their arms and legs, then all of a sudden the ability to look around becomes important. So a neck all of a sudden gains new meaning, and they already had lungs. We knew that, based on work of Bashfordine and many other people in the turn of the century. So
I'll give you actually sort of two somewhat related Hox questions here. So if you ectopically express Hox13, would that be sufficient to induce an extra autopod?
I wish, no. No, I wish it does, but it doesn't.
Okay, and then sort of related, so what about Hox genes that are expressed in the stylopod and zeugopod? Are they, what would they be expressed in the fish systems that you've looked at?
In the proximal, in the basal region of a fish fin. So what's interesting there is you have these structures that really don't look like the humerus and radius and ulna, right? But they have the same genetic networks. So there's something going, so basically there's a boundary between the proximal region and the distal region. The proximal region includes in us the stylopod and zeugopod. In fish, it's what's called the proximal radials. In the distal region, you have a single strip of bones in many fish, and in us, it's the hands and fingers. But that's a good question. I mean, it looks very different anatomically in that basal region, but they have similar genetic networks in acting. So there's something probably different downstream that we don't know about.
Right. Here's a question I know that's been around in the field for a while, and I've heard various answers to it. So let's see nowadays what the thinking is. Is there a reason vertebrates tend to have 5 digits?
I knew that question would come up. So there's two variations of answers to that. One is sort of adaptive, the other is developmental. The first is adaptive. Maybe there's something sort of adaptive about having 5 fingers as a maximum. It's a symmetrical appendage. You have 2 on either side of a central digit that is useful for walking and so forth. The other is, if you look at the evolution of arms and legs, fingers and toes, what you see is 5 is not, there's nothing really canonical about 5. We tend to look at 5 like it's special because it's us. But the reality is many critters have fewer than 5, right? Think of horses and other creatures like that. And some creatures in the fossil record have more than 5. So there are some marine reptiles that have as many as 6 or 7 digits. Indeed, if you look at the earliest tetrapods found in the Devonian, some of these had more than 5 fingers and toes. They're between 6, 7, and 8. So clearly there's not anything super canonical about 5, although in general it does seem to be a maximum for most land-living animals. So why is it a maximum? Well, my colleague Cliff Tabin back in the 90s suggested that perhaps it's related to having 5 different genetic domains in which Hox genes are expressed. It's probably more complex than that, but there's a version of that thinking that says, well, maybe the genetic basis for the controls the number of digits is related pleiotropically to other features which are important, such as the formation of the genitalia and so forth. So that if you have mutations in the number of digits, and perhaps producing more than 5 digits, 6, 7, or 8, that perhaps the organism is less fertile, less viable in some ways. And there's some argument to that, though we do see variation for 6 digits in many creatures. So there are populations of humans that have 6 fingers, Pennsylvania Dutch and others. There are 6-toed cats, the famous Hemingway cats. So we do see that variation, but the question is there hidden differences in fitness that we don't know about? That may be true. So we don't know that. Maybe creatures that have more than 5 digits are lower, fitness in some other way in reproduction, survival, what have you.
Here's a question.
It's a long way of saying I really don't know, though. I'm speculating.
I think that's been the answer for a long time. So that's great. So here's a question that is towards the other side of your work, which is, how did you get into writing popular science?
That's a good question. How did I get into it? I had a story to tell, and the story to tell was not Tiktaalik rosea, which did feature very prominently in Inner Fish. The story to tell was really sort of comparative anatomy, comparative biology, comparative developmental biology. I always loved it, and when I talked to people about it, I'd say, hey, the bones in your jaw compare to bones in your middle ear compared to jaw bones of reptiles. And they'd be like, no way. And I'd be like, way, yeah, definitely. And here's the evidence, it's development, it's fossils. And they'd be like blown away by it. And so at some point I realized many of the fundamental aspects of science that I take, that are fundamental to me and I've been living with them for 20 years, are surprising to other people. So that became the basis for Inner Fish. And while I was writing Inner Fish, we discovered Tiktaalik. And so it became truly you're Inner Fish. But the passion for it came from, there are a couple things going on. One is I wanted to convey the sort of joy of discovery that's, inherent in science. All science involves discovery, surprise, failure, learning from failure, and so forth. The other piece was, in 2005, when I began to think about writing Inner Fish, intelligent design creationism was kind of on the rise around the country. And so I wanted a book that entered that space, but not in a confrontational way, in a way that says, look, here's the science, here's what it is, evolution, all the evidence we apply, has happened, and you're a fish. So that was the idea behind it.
Here's another question here that relates to the fossils that you found. So it says the flat-headed fish looks like an alligator or a crocodile. What is the nature of the relationship between the fish and the gator?
Yeah, so this is much more primitive, if you will, than a crocodile. Crocodile and an alligator, those are reptiles. They're amniotes. They're distant in the tree of life. The thing about it is, this strategy of having a flat head with eyes on top is something we see again and again in aquatic creatures in the evolution of fish and tetrapods. And the reason is it's a really great strategy. It's a great strategy for a critter that lives on the bottom of the water column and needs to look up for prey. It's also a great strategy for a creature that's at the interface of water and land. Think of like a crocodile, where they're looking out, but they're also still in the water. So this is something we see in many amphibians. We see it in fish like Tiktaalik. We actually see it in alligator gars. They've evolved it independently. So it is a strategy that has evolved independently numerous times. And in each case, it's really kind of about the placement of the eyes, the biomechanics of the head. These things are really good snappers. They're really good biters. And so it's part of that for the biomechanics as well. So there's lots of reasons why we see flat heads, but it was definitely a big shift in the transition from, so fish originally, which had conical heads in our lineage, to ones that had flat heads with eyes on top. And so Tiktaalik is one of a group of critters. We now know several of them like Tiktaalik that had heads like that.
Here's another one about evolution of body plans. So person asks, besides the limb body plan that is ancient, the body plan of the head seems very old. So nostrils are always at the tip of the head, eyes posterior to the nostrils. What's the significance of this arrangement?
Yeah, well, it really wouldn't do to have all that stuff by the anus, that's to be blunt about it, right? There's definitely a forward piece of this. And so we tend to have much of in our lineage, at least, much of the sensory information is at the front end of the animal. There are lots of animals where the sensory information is throughout the body, lateral lines and so forth like that. But in us, in tetrapods in particular, it's front loaded. So paired sense organs are eyes, ears, nose, tend to be all clustered there because that's where the action is. And the other end has another purpose. Yeah.
I think a good example of like changing that is like whales and things like that, right?
That's right.
Yeah. Here's one about, let's see, sorry. So you spoke about genetic networks regulating the formation of the limb. However, you showed us that expression pattern of the expression pattern of individual gene. Have you looked at downstream targets of HOX13 and how similar they are between vertebrate species?
Yeah, we have. And we've also looked upstream as well. So a lot of our work has been on sonic hedgehog, GLE3, which are factors which are upstream of them. But remember, these can be upstream and downstream. So these things are expressed in multiple phases in development, and it had different roles. So they may be upstream in one place and downstream in another. But yes, we've been trying to decipher those networks quite a bit. And in fact, that's the HOX mutants have been really helpful there, because we, comparing them to the wild type and looking at the transcriptomes, we can begin to see what the HOX networks really look like. That's one area where actually the shutdown has affected Tsuya Nakamura. When he left my lab, that was one of the things he's been doing. I believe that's affected Tetsuya's work. The lockdown has affected it pretty big.
Okay, here's one more. So do Lophius anglerfish walk on fin rays, or is there more to their fins?
Both. They do fin rays as well as radials. So what you'll see is critters, fish that walk, tend to have a convergent kind of palm area that hits the ground. So in some of them, it's fin rays, where they'll have little joints between the fin rays. Others will elongate their proximal radials. In no case do they have anything corresponding, let's say, to a stylopod, zooglopod, and autopod. It's just a different, expression, a convergent expression of that walking. But it's really fascinating what they're able to do. I mean, what they do is they have very complex musculature, that allows them to control and feather the fin in certain ways that they can, contact the ground. Many fish that, that do walk also walk in mud flats. They'll breathe air through their skin or gulp air through vascularized, pharyngeal cavities. So, fish do everything. They fly, they climb, they walk from pond to pond. They can live, mud skippers can live 24 hours out of the water in the mud flats. So yeah, it's really remarkable how fish play with this space between water and land. We tend to think of them as discrete, but really the environments themselves have intermediates, in between water and land. It's not a discrete difference.
Thanks. Yeah, so we've actually got quite a lot of questions here, but we're sort of running out of time, and we'll try to pass these questions along to Neil. But why don't we go ahead and ask a final one here? So what about going from bones to movement? So, right, the appendage and the digits and everything are more than just bones. So how do you do all of that? How do you get the right muscles and nerves during evolution?
What's really fascinating is if you look at the sort of the biological, the neurobiological oscillators, the pattern generating systems, it seems like the ability to generate alternating gaits is primitive to tetrapods and probably primitive even to sarcopterygians like Tiktaalik. We see the ability to walk on alternate gaits and control distal fins in skates, in sharks, in various types of ray fin fish. We see it in lobe fin fish. I mean, this is something that's fairly general. So I'm thinking the neurobiological substrates already existed before the bones did their thing, because they were being used for alternate gaits. So alternate gaits can be used for swimming. Controlling distal appendages can be used in swimming as well, also for bottom walking. So I mean, this may be naive of me, but I think of that there's already a template written in the central nervous system.
So again, things are more ancient than you think they are.
Lillian Hillman again. Yeah, what can I say?
Yeah. All right. Well, Neil, thank you very much. I'm sorry we couldn't get to all the questions, but like I said, we'll pass them along. And thanks everyone for listening, and Neil for a great presentation.
Thank you.
Thanks.